Welcome to Brain Ponderings, your window into the world of neuroscience research, conversations with scientists at the forefront of their particular area of neuroscience research. Today, it's my great pleasure to welcome Liqun Luo. He's a professor of biology and neurobiology at Stanford University. He has been doing a lot of work over the last, 4 decades maybe, on working to understand the mechanisms, cellular signaling mechanisms, molecular codes, if you will, for the formation and modification of neural circuits in the brain. He's worked both on invertebrate systems, Drosophila in particular, where the genetics are really powerful. The nervous system is a little bit simpler and maybe easier to get it to understand the basics. But he's also done some work on mice. And so we're going to talk to him today about his work, and I'd like to welcome him.
Thank you, Mark, for your introduction. Very nice to participate in your podcast.
Okay, you're originally from China. I see you at Shanghai, I guess.
That's right. I was born in Shanghai.
And you started out there in molecular biology, I guess.
Yes, I was an undergraduate in the University of Science and Technology of China, where the major was molecular biology.
Okay, and then you decided to come to the United States. And so how did you go about that? Right, you applied to a number of different, I guess, neuroscience programs or prominent institutions and then select one, or how did that?
Yeah, no, it's actually it's an unusual path. At the time when I was college graduate, about to graduate from college, China was just opening up to the world, and there is very little knowledge about outside. So, for example, I don't know how graduate school work and all of those.
Ah,
TOEFL, GRE, nothing. So at that time, we participated in a program called China-US Biochemistry and Molecular Biology Examination and Application. So this is a program where about 60 universities joined. They give us exams in China, and then they had two professors came to came to interview us, and then each of us will assigned for four universities to apply, and we were asked which universities do you prefer to go, and I only knew two universities at that time. One is Stanford, one is Harvard. So I put both of them up, and neither of them accepted me. But the other two university accepted me. One is Brandeis, one is University of Utah. So I selected Brandeis University and ended up going there. And the reason I select Brandeis University was because I had a professor who was teaching us English, and I have no idea. I just went to him. He's from America, and he said, "Oh, you should go to Brandeis because it's, in Boston, it's a nice place for academia, things like that."
okay, and there your PhD dissertation advisor was Kalpana White.
Is that right? That's right.
And what was your project?
Oh, it's again some accident. So I was supposed to be working to identify a gene that regulates neural development. Um, and at that time, the gene was known to have a mutant phenotype, and we were doing chromosomal walk from one locus to another to see, try to clone the gene. And I didn't end up working on the gene that I wanted to work on, but in the middle of this walk, I found a gene which is expressed in nervous system, which we thought was the gene I was looking after. And it turns out that it's a gene that homologous to human amyloid precursor protein that was just discovered a year earlier to be, important for Alzheimer's disease. So at that time, we were all surprised. Why should the Alzheimer's disease gene be in the fruit flies? I was working in the fruit fly nervous system. So I ended up working on that gene and initially thought I was working on the gene I wanted to work on, but it turns out to be completely different. But its gene is interesting enough that I've focused. So I learned a lot of Alzheimer's disease.
Yeah, and my lab was focused a lot on brain aging and neurodegenerative disorders.
Yeah,
So back in the 90s, uh, when the amyloid precursor protein gene was cloned and then the amyloid beta peptide sequence was identified. And then most of the focus has been on, the pathological
yeah,
consequences of
yeah, yeah.
the amyloid, but the normal function of this amyloid precursor protein it hasn't been firmly established yet. My lab
Uh-huh.
showed that the secreted form of amyloid precursor
Uh-huh.
protein released from the cell,
Yeah.
it can uh, have a rapid inhibition of postsynaptic neurons.
Uh-huh.
Yeah,
Okay.
yeah, and so we
That
had
was in 1990s, you said.
we had a Nature paper in '96 where we did
96. Yeah, that's post my PhD.
yeah
I finished the PhD in 1992, and
yeah
at that time, still don't know too much about what the gene does. But
yeah
what we were able to show is that in the flies, if we knock out the gene, there are some behavioral phenotypes, and we can rescue it with a wild type transgene of flies, and also rescue it with the human APP.
yeah
So showing whatever the function it is, it's conserved.
yeah interesting. And did when you knocked it out,
Yeah.
did the flies have like hyperexcitability of neural networks?
To be honest, we didn't test that.
I
We
didn't
did,
say.
yeah, we didn't. We were not a neurophysiology lab, so more
Okay,
molecular genetics.
okay, let's move on. We're only at your PhD. So then you went to from the East Coast to the West Coast to San Francisco
Yeah,
with Yunong Jan. Uh, uh, him and his wife are both very famous
yeah, yeah.
neuroscientists. What did you do there?
Yeah, there I actually started to study neural development. That was the project I was supposed to work on. Um, and I was particularly interested in studying how neurons acquire their complex morphology, dendrites and axons, and how neuron from a, simple cell develops these elaborate processes. So I started to use genetics to look for mutants screenings and also did some experiment on candidate gene approach, taking an inspiration that axon growth might be similar to budding of a yeast.
Hmm.
And we know a lot about yeast budding, and some of the molecules are conserved. And it turns out that latter project works out well. We cloned, I showed that some of the genes that are important, small GTPases, important for yeast budding, play important role in axon growth, RAC, GTCDC42, these things like that. And turns out that they are important for regulating actin cytoskeleton in response to extracellular cues.
Now, I did in my postdoc with Stan Cater, I did a lot of work looking at how calcium regulates growth cones, first in identified neurons from snail, then embryonic hippocampal
Mhm.
neurons. And so the actin cytoskeleton, you have these polymers of actin, they can grow very quickly and like you have extend filopodia from a growth cone,
Right,
which kind of
right.
reach out and maybe touch a potential uh, synaptic partner. Now, what's the relationship between these GTPases and calcium? Intuitively, there must be some relationship.
Yeah, that's a good question. The GTPase work and the calcium has been largely in parallel.
Yeah.
People have not established a causal, like a linear path, which one is upstream, which one is downstream. And it's possible that they are largely parallel. Um, um, yeah. So because we did not have, for example, the GTPase work did not have any component that seems to be calcium dependent.
Hmm. Okay.
You have regulators, upstream regulators like those gaps and the gaps that are directly talking with receptors, and downstream
Mm.
with cytoskeleton, acting modulators,
Yeah.
regulating acting polymerization, depolymerization. Yeah, so that's an interesting point that these things has to be, there is more multiple roads to Rome, I suppose.
Yeah, certainly when you experimentally induce calcium influx, it can have dramatic effects on the cytoskeleton.
Hmm.
Um, okay, so you have these neurons, they're born from stem cells and then they grow
Yeah,
out neurites, and one of them often grows faster than the others and
yeah,
becomes the axon. The others tend to grow more slowly and branch more. It depends on the neuron, but
yeah,
in general, they become the dendrites. And this polarity is maintained apparently throughout the life of the neurons. And then, so what are some of the signals, right? Because you have, first, why don't you talk about Drosophila and the advantages? I kind of briefly mentioned one, the genetic
right,
aspect,
right.
but talk about their nervous system a little bit
Yeah,
and why the fly is very powerful in getting a lot of data, and not only that, but getting it pretty quickly.
yeah. So the fly, is chosen uh, as a genetic model organism because it's fast reproductive cycles. Every 10 days, you
Yeah.
get a bunch, flies can make new flies. And these are dream for geneticists that, in fact, a lot of the genetics uh, concept like genes or chromosomes all coming from studying the fruit fly from Thomas Hunt Morgan, in the beginning
Yeah.
of 20th century. So people have been using the tradition, we have mapped all the genes. There are many ways to perturb the nervous system, and we can not only create genes, but also studying their function with very sophisticated methods. For example, one of the methods that we develop called Markham that labels knockout genes in individual cells and then visualize only that mutant cells. That turns out to be very useful.
Yeah.
The original incentive for doing that was to study these GTPases. That's important for every cell, but we wanted to study what happens when you get rid of them only in neurons,
Yeah.
not in surrounding cells. And then by using this method that allow us to knock out genes in an individual cell and visualize the mutant cell, suddenly we see open up the new world. In fact, not only studying these genes, but also many other genes, or just look at the wild type, individual cells, you can label them, you can visualize, trace their axons, look at their dendrites, and that actually led me to my next adventure. The growth cone signaling was the first chapter in my research in my lab at Stanford, started with a postdoc. But then probably about 5 years into it, I started to work more into circuits, how neurons communicate with each other to wire precisely. And that's been a subject that has been starting, I would say, about 2000, the last 25 years.
Now, there's a lot of different signals that have been identified that do affect the growth and formation plasticity of synapses, neurotransmitters, neuromodulators, neurotrophic factors, cell
Mhm.
adhesion molecules,
Yeah.
and undoubtedly there's a number of different pathways that haven't even been discovered. So how do you go about figuring out what to focus on and how you get to really I guess there's a lot of different theories and potential mechanisms
Hmm. Hmm.
whereby only a few signals can lead to the formation of complex circuits.
Yeah.
Um, so, I mean, that's a big question, but
Right. We've been focusing on particular question called wiring specificity. So the study of this can be traced back to Roger Sperry's chemoaffinity hypothesis based on his observation that regenerating retinal ganglion cell can target brain target in a very precise point-to-point manner. Sperry speculated that cells in the axons in the brain must have the ability to recognize their targets with exquisite precision down to at individual cell level. So, and schema affinity hypothesis, as you probably know, has inspired generations of developmental neurobiologists to look for genes that guide axons to their targets with a lot of success. But still, to this date, we do not know how these different cell surface proteins work together to assemble a functional circuit. That's what we are after using the fly olfactory circuit as a model in the last 25 years. And also another interesting question is, if Sperry were correct that every neuron needs to be held apart from every other neuron by these cell surface proteins, we know now that human brain has 100 billion neurons making 100 trillion connections, but the human genome has only 20,000 genes. Only 3,000 of them are encoding cell surface proteins. How can 3,000 molecules specify, 100 billion neurons connections? So that's another question which we want to address in the wiring of the brain. How animals use limited molecules to wire a much larger number of neurons in the circuits. These are the overarching questions we started to work on in flies, and now in the last, quite a last 15 years or so, I kept a parallel working flies and the mammals. Flies more discovery, mammals to apply the principles learned from fly to see whether the same is true or not. And in fact, sometimes we learn new things in mammals. Sometimes we feed back to flies. So it's a very nice comparison and contrast.
So then, on top of Sperry's idea of like a one-to-one molecular code
Yeah.
uh, for the wiring is evidence, which is substantial, that there's an activity dependent aspect to it
Yes.
too. That, for example, just do a simple thing. You can culture neurons in a dish. My lab did a lot of primary hippocampal neuron cultures, and you can and these neurons will grow, form synapses. But if you block activity in all the neurons, say with a sodium channel blocker like tetrodotoxin, that has a profound effect
Uh-huh.
on the formation. Or you can use other ways to block activity in only a few neurons and see the effect. So somehow there has to be an integration of the the trophic theory of neural connections with
Yeah.
the molecular code theory.
Yeah. Yeah. So that's right. So I should say that these activity-dependent wiring neurotrophic theory are more applicable to the vertebrate. In flies, there is also activity-dependent wiring, but they play a less prominent role, I would say. The
Uh,
molecular card wiring plays a more important role. And of course, molecular part wiring also is very important in wiring the mammalian brain. At least the coarse blueprint is specified by molecular codes. And then you have on top of it activity-dependent wiring, neurotrophin, etc. And in fact, we have done some of the studies. I'm mostly a studying hardwiring molecular recognition, but I also have done some experiments exactly on testing activity and testing neurotrophin in the mammalian brain, utilizing another method that we developed, sort of learned from flies. We developed a method called MADAM, which is essentially in a mouse doing the same thing, knocking out a same single gene in a single cell and visualize its axonal dendritic morphology to allow us to investigate. Like if you want, I can give you the two examples that I
Yeah.
did on regulating using this to study activity and on neurotrophic.
Yeah, go ahead.
Okay, so maybe let's talk about the activity first. One of the key readers of activity in wiring the brain is the NMDA receptor. So NMDA receptor is a interesting, it's a glutamate receptor. So it will be, it's an ion channel. So glutamate binds will open up the ion channel. But unlike the conventional glutamate receptor, which is the AMPA receptor, glutamate binding will open the channel to allow ion to flow and depolarize the cell. The NMDA receptor is an AND gate. It requires both glutamate binding and intracellular depolarization. So it's very essential for example, activity-dependent learning, memory, and also activity-dependent wiring because it's a coincidence detector fit these Heb theory that presynaptic axon need to activate the postsynaptic neuron in order for their growth of the synapse to happen. So this is all well known, etc. But the question is, what does NMDA receptor do in the wiring of the brain? In the experiment that we did in 2000, we published in 2009. Prior to that, people already knew, for example, if you get rid of NMDA receptor, mice will not born because will not be born, will die shortly before birth because NMDA receptors required for wiring of the very important brainstem, functions. And then people can use conditional knockout to delete them only in the cortex and see what happens. And they found there is a very dramatic effect on a particular area that is receiving the connection from the whisker pad. So whisker, you have mouse has discrete whiskers, and they send a map to the cortex. It's called a barrel. It's like a discrete ball-like structure. Each of this barrel represent one whisker. So it's a one-to-one-to-one connections. So long story short, without NMDA receptor in cortex, you have no whisker, no barrels. That this discrete formation becomes all continuous. But still, people don't know whether this is caused by NMDA receptors function in an individual cell or it's a network effect because all the neurons are without NMDA receptor. So in this experiment, we did a knockout of NMDA receptor in a single neuron in layer 4, where it receives input from the thalamus that receives input from the whisker pad. And normally, a thalamocortical axon, these layer 4 neurons normally will send their dendrites into one of the barrels so that it will respond to flicker of one whiskers. And in our knockout experiment, when we could get rid of NMDA receptor in a single cell, because we are only affecting one cell, the growth pattern is normal. But that cell, if you look at the dendrites, instead of going to one barrel, it's spread around multiple barrels. And it turns out during development, there is a period when the dendrite spread from multiple barrels. And then through this activity-dependent refinement, they focus on one barrel growth and then prune the rest of the barrels. So if you don't have NMDA receptor in that cell, you cannot do that. So
And
that
it's not,
shows a cell autonomous function.
and is that the same thing that happens if you cut the whiskers off the animal so that it can't feel when it's
Yeah, if you cut the whisker at a critical period before the first 5 days, the barrel don't fall.
yeah, yeah. Now, it, yeah, my main discovery in my postdoc was that glutamate plays an important role in the formation of synapses in the developing hippocampus.
Uh-huh.
And that at the same time, that was shown by Martha Constantine Patton and Holly Klein
Right, right.
in the frog visual system. And that's one of the reasons why I focus so much on calcium.
Uh-huh.
And of course,
Yeah, NMDA receptor conduct calcium, of
it's a
course.
calcium channel.
Yes.
So we come back to that. Okay, so this is really elegant
Yeah. And
technique.
the other example, maybe if you let me
Yep,
continue about neurotrophin since you mentioned it.
yep,
And the neurotrophins are these factors that are produced classically by postsynaptic cells like muscles or to keep the neurons survive. Okay. And that most of this work was done in peripheral nervous system, in the sympathetic ganglia, in sensory ganglia, etc. The role of their function in the central nervous system is unclear because each of these neurons make connections with multiple targets,
yeah.
whether these play a role. So in that regard, we actually utilize the same method, deleting the gene that encode a neurotrophin receptor called track C, which is responding to neurotrophin 3 in a single cell individually. And we find that actually cells don't die. But one thing which is striking is that when we look at the whole brain, in particular in the cerebellum, the dendrite don't grow. They grow as half as much as the normal dendrite next to it.
And that's
And
the big Purkinje neurons you're
yes,
talking?
the Purkinje neurons.
Yeah,
That's
yeah.
right. And you can say, okay, maybe the neurotrophin receptor is important for growth of the dendrites, but it's actually more interesting than that. If you delete neurotrophin receptor in all Purkinje cells, they all grow fine. Only when you deleting a single one and the neighboring is good, then it's bad. So it shows that there is a competitive process in the
Huh.
dendritic growth that's facilitated by this neurotrophin signaling.
And is there a corresponding change in the number of synapses received by the?
Of course, if
Yeah,
dendrites grow as only half as much, they
yeah.
only receive half as many synapses.
Okay, and then the animal, does that have lead to a like deficit in motor function?
No, no, because we are deleting them in a very small number of cells.
Oh, okay. All right. Yeah, yeah. Yeah, those neurons are fascinating.
Yeah,
So huge, they're huge and elaborate, complicated. They're also, this gets to the question of those are inhibitory neurons. It's the cerebellum's kind of interesting because the main output of, by the way, the cerebellum has about as many neurons in it as there are in the rest of the brain.
actually more.
More.
Cerebellar granular cell alone is like 60% of all neurons.
And those are excitatory
Yeah,
glutamatergic
those are the
neurons.
inputs to the Purkinje cell.
Yeah, so the output, it's interesting, the motor system outputs of the cerebellum and the striatum are both inhibitory. It's,
Yeah,
the way this evolved is interesting because you've got the upper motor neurons that are like constantly sending signals for you to move.
yeah, yeah.
And and these inhibitory neurons in the striatum and maybe cerebellum are kind of making sure that only the
Right
appropriate,
move is
only,
made.
yeah,
Yeah,
yeah, interesting. So what about okay, let's get to some of your more recent stuff just in
yeah,
the last 4 or 5 years. And
yeah.
yeah, I put down here four, and these are all in the top journals: Neuroscience, Nature. Um, yeah, let's start with this one. Dimensionality reduction simplifies synaptic partner matching
Yeah.
in the olfactory circuit. And by the way, this is the olfactory circuits are an area you've focused a lot on, at least
That's
in
right. In the last
the,
25 years, as I mentioned, in the
yeah,
flies.
yeah.
Yeah, I can give you a general outline of what we did in the 25 years, and
Okay.
then
All right.
focus on these recent studies. Okay.
Good. Good. Yeah.
So in olfactory, the reason we love the olfactory system so much is that its simplicity and elegance. So odors are smelled by neurons in the peripheral called the olfactory receptor neurons. or ORNs. Each ORN expresses one receptor. That's the same in flies and in mammals. One, maybe two, not more. And then neurons express the same receptor, project their axons to the same spot in the brain. In a mouse or in a mammals or vertebrates, it's called olfactory bulb. In the flies, it's called antenna lobe. And these ball-like structure called glomerulus is a collection of all the axons that come from the same neurons expressing the same odorant receptor and therefore smell the same odor. So essentially, the brain creates a odor map using these balls to tell you, okay, if you are smelling banana, these three balls gets activated. If you smell apple, that three balls, maybe one overlap with a banana, gets activated. So this combinatorial activation of these glomeruli allow the animals to know what kind of odor the animal is experiencing. And then this information from these olfactory receptor neurons gets transmitted by a second order neuron called projection neurons in the fly, in a mouse or vertebrates called mitral cells. These neurons send dendrites into one of these ball, and one projection neuron sends dendrites into one of the 50 ball-like structures. And therefore, essentially, you create a 50 types of olfactory receptor neuron match precisely with 50 type of projection neuron in these 50 glomeruli in a 3D structure that is essential for odor representation and odor interpretation and odor-mediated behavior, whether it's innate or learned based on downstream targets of these projection neurons. So because of this, we were focusing on how these 50 partners match with each other. And the other beauty of this is that usually if you want to study synaptic matching at a synaptic level, you really need to look at EM, because the synapses are small and the light microscopy cannot resolve individual synapses easily. But then the olfactory system essentially is a biological amplification of synapses, because each of this ball is 1,000 synapses, but they are the same kind, representing the same odor and picked up by the same projection neuron, the same kind. And next one is a different kind. So based on and these balls are like 5 to 8 microns big, so it's much easier to visualize in light microscopy. So we can study synaptic specificity using just a light microscopy. So it's natural amplification for us. So we've been studying this for the last 25 years, as I mentioned, and studying every details of how this one-to-one matching happens during development, using sophisticated tools in fly genetics. Sometimes we invent them, many of them. So a short story is that in fact, this process is a multi-step process. In the beginning, actually, it's the second order projection neuron themselves. They send dendrites into this proto-antenna lobe, and that dendrites sort themselves out into different groups before the olfactory receptor neuron axon even enter the scene. And then olfactory receptor neuron enter the scene. Usually they are moving on the surface of this three-dimensional structure. And then they start to recognize their partners. ORN1 connect with PN1, ORN2 connect with PN2, ORN50 connect with PN50, etc. Right? All very orderly. So that we made a lot of progress in identifying what kind of molecules may be involved, etc. But still, coming now back to the dimensionality reduction study that I mentioned to you. So finding one partner out of 50 in a 3D structure is actually pretty complicated task. You need to navigate in 3 dimensions, you need to know where to turn, etc., etc. But it turns out things are simpler than that. I mentioned to you that our axons first grow on the surface of this ball. It turns out these dendrites, they are sticking out their processes. Every one of them go to the surface, even though the dendrites, the final target could be inside. But during development, they stick the dendrites out to meet incoming axons. So that 3D recognition become a 2D because surface is easier. And then there are more patterning. ORN axons, even though if you look at it collectively, they cover about 270 degrees of the ball. But if you look at individual axon, individual type, they actually move along a very narrow arc, about 30 degrees, which matches precisely
Huh.
with the dendrites of their postsynaptic partner. So essentially choosing one out of 50 in a 3D becomes choosing one out of maybe a handful, 5 to 8, along a 1D trajectory. So it's a much simpler problem. So
Wow.
that's the essence of that dimensionality reduction
Yeah,
study.
that's really a fantastic finding. And do you think the same general mechanism of dimensionality reduction applies to vertebrate?
Oh, yes, yes.
Yeah.
Um, there are actually naturally occurring dimensionality reduction. For example, in the retina, you have layers.
Yeah.
So, you go to the right layer, you find a 2D, and then you can
Yeah.
reduce it. For example, in the cortex, your left side of motor cortex will project to the right side of motor cortex, right? So, and that's very precise. Not only which area, but which layer it is. And if you look at, although molecular mechanism not as known, not much is known, but if you look at just anatomy, they first go through the white matter to find the right 2D structure, and then they ascend. Instead of searching a 3D volume,
Hmm.
you find a 2D map plus 1D. So these are some of the examples we actually discussed in this paper.
Yeah. Yeah.
And maybe, if I guess the follow-up of that question is, okay, you reduce dimension to 1D. What happens? How do you still pick one out of 5 or 8
Yeah.
as a synaptic partner? So that's actually the subject of two recent papers just published a few weeks ago. One is called the one which I'm actually most excited about in my entire 25 years of studying the olfactory circuit is the rewiring. I don't know whether it's on your list. Rewiring of the olfactory circuit using a combinatorial code. It was published in Nature 2 weeks ago.
Yeah.
And that one was, really, I would say a remarkable achievement. So I told you about this one-to-one connection, 1 out of 50, and we identified many genes that we deleted, you lose specificity. But usually, half of the neurons still fine. The other half make a mistake. And when they made a mistake, they go to different places. You can say that, oh, maybe this system is very redundant. Each gene plays a role that other genes can compensate. And that will increase the robustness of the system, which is true, probably. But on the other hand, it's harder for us um, to really try to understand, because every time you delete things, something is wrong, but not completely. If you delete more things, more messed up. So you don't know where to claim that you reached a thorough understanding. So about 10 years ago, I actually challenged everybody who come to work in the lab on olfactory system. Maybe we should do a re-engineering experiment, okay? Instead of disrupt the wiring, we want to rewire. If you can rewire ORN1
Yeah.
to PN2 completely, then I could say you achieve a milestone. It's a milestone achievement because you must have figured out how do you do that, how naturally ORN1 go to PN1 but not PN2, and etc. So that was the essence of this experiment. But this experiment took a lot of other, essentially accumulative knowledge in the last 25 years, all went into that experiment of understanding how this works, including dimensionality reduction. So we know that you need to choose with along 1D.
Yeah.
But how do you choose that? Again, going back to Sperry, it must be some molecular recognition. You choose PN1, ON1 choose PN1, but not choose PN2. So it must be PN2 and PN1 has some differences in
Right.
their cell surface protein expression. ON1 matches with ON PN1, and ON2 matches PN2. ON1 versus ON2 must be different. So because of that hypothesis, we actually did so-called single-cell RNA sequencing to look for differentially expressed genes in ON1, PN1, ON2, PN2, essentially the entire olfactory system. And in principle, let's say if you change the cell surface protein from ON1 to that of ON2, of course they are going to connect with PN2. So, but that is not an easy experiment because ON1, ON2 differentially express 50 or more cell surface proteins. Even in flies, you cannot do all of these. So we have to make a educated guess of which of these different 50 genes are important. And along the way, in other studies, we find that these cells express two types of proteins. One is called homophilic attraction proteins. So ON1 and PN1 express the same cell adhesion proteins that are not expressing ON2 and PN2. PN2 and ON2 express a different cell surface attraction proteins. And so we initially found, about 12 years ago, found two of these. And now from the single cell RNA sequencing comparison, we found two more. So we have four of different pairs. Now, the other class of proteins that are very important are repulsive proteins. In addition to tell you, oh, come, you will tell your partner, come, you will tell your non-partner, stay away from me. These repulsive proteins are also very important. And from this single cell sequencing comparison, we identified 3 pairs of repulsive proteins. These are not homophilic, these are heterophilic. A repel B, C repel D, etc. So now with 10 different molecules, 4 of them matches, 6 of them repel, we can now start to play matching, mismatching things. And it turns out that ON1, we can change about 5 molecules. Some are repellent, some are attractant. 3 things you need to do to rewire. For ON1 to rewire to PN2, first of all, you need to loosen up your connection with your original partner. You add repulsion to your original partner. Second, you need to
wait a minute.
Yeah.
So you're doing these manipulations after the circuit has already been established?
No.
During development.
During development, it has to be during development. Yeah. So you increase repulsion to your original partner, and you remove repulsion to your new partner.
Yeah.
And you add adhesion to your new partner that lose adhesion to your old partner. So with all of these 3 different things together, so we can matching attraction, decrease repulsion with your old partner, increase repulsion with the old partner, decrease repulsion with the new partner. With all these 3 things, we were able to rewire in this paper, we rewired 4 different pairs to 2 of them actually complete. 2 of them like more than 50%. We still probably miss some molecules. So, but
That's
still,
you had two back-to-back papers in the same issue of Nature.
Yes. Yeah.
And then you're talking about
one of them.
All of them.
Yeah, and no. One paper is talking about identification of these 3 repulsive pairs.
Right.
Which is essential for us to do the rewiring.
Yeah.
So the second paper that I've been focusing on is the rewiring to mismatch. And one of the, I don't know whether you have seen that, after rewiring, not only we rewire the anatomical connections, but we rewire the physiology and interestingly behavior.
Yeah.
So the original ORN1 is smelling a pheromone that tells males to not mate with a male. Male mating, of course, is not very productive.
No.
So you need to inhibit that. And the recipient of rewiring is a PN that promote mating. So after we rewire anti-pheromone to a pheromone, for mating, then rewire the male will chase after other males.
Interesting, very interesting.
Yeah.
And so you can imagine you can do that for assuming the same thing's true in, say, a mouse, you could do this kind of manipulation and you could make it so a mouse would be attracted to the odor from a cat, or at least not
Repelled.
repulsed,
Yes,
or
yes, yes. But
well,
it's much harder to do this experiment in a mouse because, for example,
uh,
in that rewired mouse, rewired flies, we need to have 11 different transgenes in the same fly.
yeah,
And putting 11 different transgenes in the same mouse is going to take forever.
yeah.
And also, it takes a lot of experiments to know which five genes you want to manipulate. And so I would expect that it's much harder. But we're actually doing some of these experiments, not in olfactory system, but in some other simpler system where maybe fewer changes is required.
Would uh, would CRISPR-based approaches be of value in trying to
Yeah, it will
identify?
certainly help. It will certainly help.
Yeah,
But I think the efficiency, etc., is not
yeah,
still high enough for
yeah.
these kind of precision experiments.
Now, in mammals, the olfactory system is different in many ways, but
Yeah,
from other sensory systems, in that there aren't the connections don't go to the cortex through the thalamus.
that's right. It's very fast.
Yeah,
Mitral cell that's recipient of ORNs go directly to piriform cortex, which is the largest part of the olfactory cortex.
yeah, and that's very interesting. The there's a lot of research on uh, things getting into your nose, whether it's a
Yeah.
virus or some toxin or pollutant
Uh-huh.
or so on. And in mammalian systems, the projection from the olfactory neuron that goes to a region called the uncus,
Uh-huh.
and that's has close connections with the hippocampus, which is
Yeah.
a
Yeah.
very
So part of the, actually olfactory mitral cells, some of the mitral cells goes all the way to lateral entorhinal cortex,
yeah,
which is a direct input to the hippocampus.
yeah,
And in fact, we are studying some of those connections. Some of the molecules we identified in fly ORMP and connections. So these are homophilic attraction molecules. We find them to be expressed in hippocampal entorhinal circuits. And they are doing very similar things. And we have also found additional molecules that not in flies that play a role in regulating the wiring specificity of the hippocampal entorhinal circuits.
What are some of the specific names of these?
So the molecules that we've been working on the most is called tannurins.
Uh,
I often joke that my student who got this first identified tannurin as homophilic synaptic partner matching molecule in 2012, Wei Zehong, now has become a famous professor in UCLA, got tenured of, like 4 or 5 years into
wow.
assistant professor after he arrived in UCLA. I said, see, work on tenuring, you can get tenure. So most people remember that name now. And tenuring has in addition to being a homophilic attractant, it also has a partner called a lateral and we show in the hippocampal study that lateral and tenuring are binding partners, but they are repulsive. Tenuring can serve as a repulsive ligand for lateral expressing neurons,
Hmm.
and lateral can also serve a repulsive ligand for tenurin expressing neurons. And this mutual repulsion between tenurin expressing cells and lateral expressing cells actually play a very important role in setting up the parallel circuits in hippocampus, medial lateral entorhinal, proximal hippocampus, CA1, distal CA1, etc. So that these two parallel circuits play distinctive role. One is more for spatial recognition, the other one is more for object recognition.
Yeah, interesting, interesting. Okay, and then uh, how big is your lab now? It must be pretty big.
I have about 8 PhD students and 8 postdocs.
Yeah.
So it's pretty big, yeah, but not enormous.
Yeah, it's I had a big lab, and at some point you get to where you end up sitting at your desk all day.
Yeah, yeah, yeah.
And I kept until I retired, I would spend one morning every week dissecting out hippocampus from like 40 rat embryos.
Oh, really? You still kept your art.
Yeah, and
I have not been able to do experiments for a long time.
well, I didn't do any experiments, but
Yeah.
I tell my postdocs and students that I'm their technician.
Yes, yes.
I'm
I have been doing that in the first 5 years of my assistant professor. I was the fly technician. I make flies for them, put different genes together, things like that. At that time, we were mostly working on flies.
yeah. Yeah, and then actually nobody wanted to do what I did because I could do it better and quicker because I've done it so many years. It worked out
Right.
pretty good. Um, okay, so you're going to keep focusing then on the combinatorial codes and these.
So that, yes, we are going to work on a little bit more. There are more to be explored. We were able to rewire, you know, inhibitory pheromone to attractive circuit, but now we are trying to expand the rewiring capability. Maybe we someday will rewire a food that's detecting odor to a pheromone, things like that flies. And as I mentioned, we are trying to apply similar principles in the mammalian brain. And also, I must say that working on wiring of the brain development is about half of what we do. The other half are working on, organization and function of neural circuits. Most of these are now working in a mouse. And evolution,
And
there
what?
is also a little um, one person is working on evolution of the nervous system.
Oh, interesting. Yeah.
Yeah.
And okay, so to what extent do you think some of these same mechanisms that are involved in the initial wiring of circuits during development are also responding to environmental factors that affect that stimulate plasticity, whether it's physical exercise, cognitive stimulation,
Right, right, right.
uh, uh, chronic
Um,
stress, those
I think
kinds of.
some of these do. These physical, the stimulus induced rewiring usually happens in very small scale,
Yeah.
not long distance, right?
Yeah.
Some of those final synaptic partner selection molecules might be used. For example, the tenuring electrophiling that I mentioned to you during development in many circuits. In fact, they are repeatedly reused in wiring many circuits. But most of the circuits, these proteins are turned off. But
Ah,
in the hippocampus, somehow it's constantly on. The expression level is reduced compared to development. They are still maintain their specificity. And we suspect that maybe, hippocampus, of course, is one of the most plastic regions. So maybe it's possible that these molecules are continuously being used. I
well,
should
that's
say also that same molecules also play a role in synaptic synapse formation. So they could be playing that kind of role.
that's also very interesting based on the fact that the two brain regions were in man, mouse, and probably humans, where neurogenesis occurs in the adult are hippocampus and the subventricular zone, which is those stem cells give rise to olfactory bulb
That's right.
nerve.
That's right. That's right. At least
And,
in mouse. In humans, a little controversial.
okay. Well, it's hard to study in humans.
Yes, yes. All the evidence are more or less indirect. So that's why some people see it, some people don't. That's why it's controversial. In the mouse, it's unequivocal. Yes,
Yeah.
these are the two regions where more, adult neurogenesis contribute to the circuit function.
Right. And do you think then there's more of a in the hippocampus? Well, I don't know. Can you can you extrapolate what you found in the olfactory system to the hippocampus in terms of maybe there are specific molecular codes for individual post and presynaptic neurons in the
Yeah,
hippocampus.
yeah. I do not think. I mean, some of the molecules, in fact, the tangerines we originally identified in the fly olfactory system, they also express in mammalian olfactory system. We will actually try to study them, but it's too complicated. And then we find the hippocampus, they are nicely patterned expression matches with their connectivity. So we studied we switched to studying them in the hippocampus. So certainly molecules can be reused over and over again. But in terms of like point-to-point connection, like Sperry's original hypothesis, I don't think in a mammalian CNS
Yeah, it's too
it
many.
needs to be that precise because, like you said in the beginning, activity experience can shape
Yeah.
the refined connectivity. So as long as roughly the right brain region goes connect with the right targets in the right region,
Yeah.
the molecules may not need to do the micromanaging of, with whom, which individual neuron I connect with, how many synapses I to form. All of these can be left to, experience.
Yeah. Yeah. Okay. Yeah. And you mentioned early on how the flag has led to a lot of discoveries of genes that uh, and then play important roles in development, a lot of things, and then their homologs in mammals are discovered later, and there's a whole series of things. We worked briefly on sonic hedgehog, for example,
Uh-huh. Yes, coming from
and
hedgehog
yep,
in flies.
and in the aging field, the the insulin signaling pathway was really shown to be
Right.
important first in Drosophila because you can quickly get
Yeah.
knockout and feedback and see
Yes.
that the animal lives twice as long or whatever.
And the C. elegans, actually.
And the worm, yep.
Yeah.
Yeah, and there it's even more than Drosophila.
Hmm.
Everything's known.
Yeah.
All the lineages,
Yeah.
all the cells. Um, okay, so I guess the take-home message there, though, is that each individual neuron in a fly is more important than each individual neuron in a mammal for
Yes,
the,
we have more neurons than fly neurons.
yeah,
And each individual neuron in C. elegans in worm is even more important than each individual neuron in flies.
yeah, yeah.
Right? Neuronal number plays a role in, how you use them.
One of the tenure track investigators in my NIH lab, Kathy Wolkow,
Uh-huh.
she showed that insulin signaling pathway in only two neurons controls lifespan.
Uh-huh. Yeah, yeah.
That's pretty remarkable.
Yes, yes.
Okay, well, I've taken up an hour of your precious time, and I really appreciate you know, offering that time, given how
Yeah,
busy
it's
you.
happy. I'm happy to, education. I have spent, I wrote this textbook
Oh yeah,
in
yeah, I forgot to
undergraduate
mention
teaching.
that.
Yeah, yeah. So education is one of my priorities. In
That's
fact,
right.
right before talking with you, I just finished a lecture to Stanford undergraduate. I'm teaching them Introduction to Neuroscience
Yeah,
using this textbook.
yeah, I see from the Stanford webpage that
Yeah.
you're widely recognized for your teaching, and your neurobiology textbook is widely used in undergraduate and graduate
Yeah,
training.
I'm very happy that that's the case, because I try to teach them discovery process, how discoveries were made rather than the facts, what's known. So it's more interesting, and I'm happy to see that this kind of philosophy is adopted by other universities to teach them the same.
Yeah, that's really good. That's and your writing is just fantastic. I'm going to put in the description section of this podcast links to several of your papers and including a review article you had in I don't know where, in Science, I think in 2021 or
Oh, that's right. Architectures of neuronal
yeah,
circuits.
which is something that uh, didn't talk a lot about today, the
Yeah.
details on this. It's complicated, but there's all these
Yes.
different processes whereby
Yes.
the brain deals with
Yeah. How
information.
you build the brain from neuron, small circuits, and large circuits in the whole
Yeah,
brain. So
yeah,
that was actually the incentive for writing that text, the articles. I just finished the second edition of my textbook. Because for textbook, of course, it should go into more elementary, and for the undergraduate students, I felt I should distill some of the things I learned from writing the textbook into a article that's more sort of for professionals, not
yeah,
only neuroscientists, but also a lot of engineers or computer scientists can
yeah.
get some inspiration from the brain science, the recent advance in brain science. So that's the incentive for me to write that article.
Artificial intelligence. Yep,
Yeah.
yep. Okay, great. Thanks a lot, Li Chun, for your time
Yes.
and very interesting work. And I'll try to keep up with it. Yeah,
Yeah. Thank you, Mark, for inviting me again. It's fun to talk about brain development.
yep. Bye.
Bye.